Endomembrane trafficking is the transport system for moving proteins and molecules through the cell, as well as the interactive surface for protein synthesis. But first, let's understand the endomembrane system. The endomembrane system is the system of internal membranes with eukaryotic cells that divide the cell into functional and structural compartments, or organelles. The nucleus serves as the control center for the cell. It contains DNA, RNA, and mRNA. Cytoplasm is between the plasma membrane and the nuclear membrane. The plasma membrane is the cell's membrane. It is a phospholipid bilayer and it is amphipathic. DNA, deoxyribonucleic acid, is found in the nucleus. It is genetic info that is the main component in chromosomes. A molecule of DNA is formed by complementary nitrogenous bases. They are adenine, tyrosine, cytosine, and guanine. The endoplasmic reticulum performs biosynthesis functions and manufactures membranes. The ER connects with the nucleus and extends throughout the cell. There are two types of endoplasmic reticulum, rough and smooth. Rough has ribosomes and smooth does not. Ribosomes are where the protein synthesis takes place. The Golgi apparatus is made up of many sliding membranous discs. Cisternae are the chambers within. The cis face is where the proteins arrive and are taken in for modification by enzymes. The trans face is where the product exits, and secreting vesicles contain secretion that will be discharged through exocytosis. Cells generate new protein using DNA. This is called protein synthesis. Transcription happens in the nucleus. It copies DNA to mRNA, which is messenger RNA. Here, the double helix of DNA is being unzipped by DNA polymerase and enzymes. Translation. mRNA moves through a nuclear pore into the cytoplasm. mRNA enters the rough endoplasmic reticulum and binds with the ribosomes. The start codon allows the tRNA to come with anticodons and amino acids to make polypeptides. Anticodons are nitrogenous bases on the tRNA molecule that interact with codons on the mRNA strand. The stop codon stops this process. Polypeptide formation is when mRNA binds to a ribosome and tRNA delivers amino acids to the mRNA. Polypeptide processing begins once the mRNA arrives at the rough endoplasmic reticulum. Proteins are folded and placed in a transport vesicle. Then they are sent to the Golgi apparatus for protein modification. The protein enters at the cis face, then exits at the trans face by secreting vesicles or lysosomes. Lysosomes contain digestive enzymes, and secreting vesicles exit the cell by exocytosis. Exocytosis is the release of cytoplasmic materials by fusion of the membrane vesicles with the plasma membrane.